related to healthy aging and to centenarians. Third, his group has recruited centenarians from all over Italy. Every corner of Sardinia to the mainland, they have recruited centenarians, and there are many. Fourth, his group has created a database and a bank with materials, with a sera, DNA samples, and so on, which are very valuable in this field. And then the fifth is he has provided leadership. And what better way could we understand his leadership by reading out the title of his talk, which is Nutrition, Aging, and Longevity, the Strategy of the New European Union Project, which commences in 2011, this year. So please welcome me, Professor Franceschi.
Thanks a lot. Well, first of all, thanks for this introduction and kind introduction, and I thank you very much. And I would also like to thank the organizers, Josiane Sillard and Bertrand Friguet, among others. Well, as you know, aging is a complex trait. The aging scenario, according to what I learned this year, is that the aging phenotype is the result of two major processes. The first, everybody knows, is the accumulation of unrepaired damages at all levels of the biological organization: molecules, cells, tissue, organs, entire body. The second is something that is not appreciated too much, and is the capability of the body to react to such damages by activating a variety of molecular and cellular basic defense mechanisms in order to neutralize their harmful effects. And since it's so complicated, a systemic approach is needed to address such a complexity. The aging mosaic should deliver you this message if this piece of this puzzle are cells or organs or individual or organelles, and the black dot are the scars, the damages which accumulate in different organs and cells. We are a mosaic because every cell, every organelle, every tissue accumulate damages at a different rate. So there is a different rate of aging, and eventually we are like we start like a fine-tuned orchestra of different organs and cells, and we end with an orchestra which play in a way which is not particularly good. I think that omics, today we have the possibility to use omics, high throughput technologies, and so a comprehensive integrated approach. The models that we are using in the most important models we are using in my lab are centenarians and their offspring, 90 plus C pairs for familial longevity, Down syndrome subject as an example of accelerated aging, monozygotic and dizygotic twins to disentangle genetic and environment, of course adult and all subject of normal aging, and recently we are starting a research on newborn children because the hypothesis is that a part of the aged phenotype is decided at early age and probably also in the 9 months in utero. And now to the project. This is the email that I sent on May 13, 2010, say about 1 year ago. Dear New Age partners, Gaudium Magnum Nuntia Vobis, Habemus Projectum Approvatum. As you can see from the attached document, our project got 14 out of 15 marks, and as far as I know, it ranks first among the 17 projects that have been evaluated on this call. This is a great success. Let me thank in particular the members of the EU office in Bologna who supported us until the last minute. And indeed, I remind you that the project was sent by email 4 minutes before the deadline. And this is the British comment of a friend. Do you think, Claudio, that if you had carried on drafting and improving the proposal until 2 minutes before the deadline, we would have been scored 14.5 out of 15, or even 15 out of 15? I think that I think that this is a good comment. This is a project is on KBBE and the Food, Agriculture and Fisheries and Biotechnology. The full title is New Dietary Strategies Addressing the Specific Need of the Elderly Population for Healthy Aging in Europe, and the acronym is New Age, Nutrition and Age. It is a big project, 16 European countries, 14 research institutions, 5 SMEs, 9 enterprise, 3 stakeholders, for a total of 31 partners. These are some of the partners, Bologna, East Anglia, and so on. These are the other one. And Italy and France are leaders for components each, and then all the other, Belgium, UK, and so on. The basic idea of this project is that nutrition can impact on inflammaging. The idea is that an appropriate whole diet, an ad hoc fortified Mediterranean diet for the elderly, can decrease the level of the chronic, subclinical, low-grade inflammatory process characteristic of old age that I have proposed to call inflammaging. Inflammaging is a major component of immunosenescence, as you remember, but inflammaging is very complex, is local and systemic, and much more complex than we previously thought. And indeed, there is still an unsolved problem of biomedicine, which are or are the causes of inflammaging, and where do the inflammatory stimuli come from. Of course, there is a genetic basis, there is a genetic predisposition to inflammaging. These are some of the SNPs and genes that we studied in the last year, which suggest that there is a genetic component. Another major contributor are persistent infections, what we call the antigenic load, and infections such as cytomegalovirus, EB virus, and so on, contribute to inflammaging. Another contributor are the senescent cells. When cells accumulate unrepaired damage, they become senescent, they stop the cell cycle, and in order to stimulate tissue repair, they produce pro-inflammatory cytokines, and their secretome is an inflammatory one. And of course, the stop of cell cycle is good because it's anti-cancer, but the production of pro-inflammatory cytokines is a strong activator of cancer progression, as you know. Another point is, as you heard before, that the fat mass produce a lot of inflammatory cytokines. During aging, we lose muscle mass and strength, so it is called sarcopenia, and increase fat mass, and macrophage invade the tissue, the fat tissue, and produce large amount of inflammatory cytokines. Another important point is the gut. You know that there is the gut is extremely important, and a lot of attention is now being paid to the gut. And there is, for example, a gut-brain axis, so signal from the brain go to the gut and vice versa. And We have the idea that inflammation is systemic, that the liver, the immune system, the adipose tissue, the muscle, the brain, the gut microbiota, and the gut immune system are involved. In particular, we are speculating on the basis of published paper that inflammation is particularly important for the brain, and that the neuroinflammation and neurodegeneration can have a strong, of course, inflammatory component, and that the gut microbiota can contribute to this. Why is it important to counteract inflammation? Because the major age-related diseases share an inflammatory pathogenesis. Atherosclerosis, cardiovascular diseases, metabolic syndrome, type 2 diabetes, osteoporosis, Alzheimer, cancer, major depression, sarcopenia, frailty. So if we are able to counteract the increase of inflammation with age, we could reduce the risk of all these major diseases. New Age will recruit subjects from 65 to 79 years, a total of 1,250, categorized as non-frail or pre-frail in 5 European countries, Italy, France, UK, the Netherlands and Poland, and will divide it into 2 groups. One will follow a whole Mediterranean ad hoc fortified modified diet, and another will continue its own diet. At time 0 and after 12 months, a variety of parameters will be measured. I have no time to go into the details of the pyramid that we have envisaged for the elderly, but there is a lot of water, not so much, a lot of fruit, vegetables, and some pasta, not much, some wine, and so on. But I can give you details. Of course, we will do during the New Age a lot of genetics, and the genetic analysis will be performed in all the recruited subjects in some of the candidate genes that the literature and our own work suggest be involved in inflammation. Another point is that we will do a lot of epigenetics, because nutrition is known to produce epigenetic changes, and all the recruited subjects will be studied for DNA methylation at candidate genes involved in inflammation. We will do also genome-wide epigenetic study, but I will talk this later. Immunology, all the subjects will be assessed for seropositivity, plasma level of a variety of important cytokines and other mediators, homocysteine, folate, vitamin B12, and so on, telomere lengths. And New Age omics. In a subcohort of people, 125, before and after, we will do an in-depth immunology study, transcriptomic on peripheral blood cells, oxolipidomics, metabolomics, gut microbiota assessment, and whole genome epigenetics. These are some of the in-depth immunology, which will be done in IFR, UK, expression and responsiveness to toll-like receptor, plasma level of inflammatory and regulatory cytokines, and so on. One of the basic ideas is that the effect of all diet is mediated by the effect on the gut microbiota. And the gut microbiota has a pervasive systemic influence on a variety of organs and systems. And in order to understand this, the New Age rationale and main goals, I will briefly summarize some recent data that have been obtained in my lab. I'm performing studies on the gut microbiota metabolomics with the people in Bologna, Patrizia Brigidi group, Willem de Vos in Wageningen, and the people at Nestec in Lausanne, Sergio Ressi and Sebastiano Collino. You know that the bacteria in our body have been called invisible partners. They are a lot present in the colon, but in the mouth, in the esophagus, the skin, stomach, vagina. And they are a lot, from 10 to 100 trillion, for a total of several thousand genera and about 16,000 phenotypes. And their collective genome is called microbiome and contains 100 times more genes than our human genome. And each human has a metagenome, say his own genome plus the microbiome. And we have to consider a Homo sapiens not a single animal, but a superorganism. And of course there are Firmicutes, Bacteroidetes, Proteobacteria, and so on. These are the main categories. The main function of the gut microbiota is protective against pathogens, trophic for epithelial cell proliferation, development, maturation of the gut immune system, and has also metabolic and nutritional role, fermentation of non-digestible dietary residues, salvage of energy as short-chain fatty acids, and production of vitamins. We have used for our study the HIT chip, the human intestinal chip. You know that in all the bacteria, the small subunit ribosomal RNA is present in all, and includes about 1,500 base pair, and is highly conserved in evolution, and contains several hypervariable regions that can be used for the identification of specific microbial species. Each chip contains approximately a little bit less than 5,000 probes, about 5 or 6 per bacteria, and so we can identify more than 1,140 targeted microbial phenotypes. This is a paper that we have published recently in the gut microbiota in centenarians and their offspring, and we studied 21 centenarians, their offspring, control for this offspring. The offspring of centenarians are about 70 years old, so we have another control group of 70 years not born from centenarian parents, and a group of young people. What we found is that in centenarians there is a decreased diversity of the gut microbiome, a decrease in Clostridium cluster 14, this is a butyrate producer, a decrease of bifidobacteria, which are good bacteria, and a rearrangement of Clostridium cluster 4, which is again is a good butyrate producer, and an increase of pathobionts, so the bad bacteria. This is a heat map of the microbiota of the centenarian, of these people that we studied, and you see that despite they are different centenarians, 15 out of 21 cluster together, suggesting that they have their microbiome is similar and peculiar. In particular, we have shown that about 9% of the total variability of the gut microbiota can be related to the pattern of the inflammatory cytokines. All these people have been thoroughly studied and immunophenotyped, and you see that you can see that the centenarians have high levels of IL-6 and IL-8 in comparison to the young and the old controls, and you can see also that the centenarians here are quite apart from, separated from the other two groups, and these are the group of bacteria which correlate with IL-6 and IL-8, two strong inflammatory mediators. So the major finding was that the gut microbiota is stable throughout adulthood if you stay in good shape, in good health, and its stability may last longer than expected. Changes do not follow a linear relation with age, because after age 70 they start changing. An aged gut microbiota occurs likely after age 75 or 80, again if you are in good shape. Gut microbiota composition of parents and offspring show no significant similarity, suggesting that the environment play a major role, and there are geographical variation. In fact, a paper that has been published after our paper on Irish people suggests that there are differences between Italian and Irish, despite the fact that both are Catholic. The gut microbiota change in centenarians is accompanied by an increase in pro-inflammatory cytokines like IL-6 and IL-8, suggesting that, supporting the hypothesis that the gut microbiota contributes to inflammation. Now what we are interested in is to look at the metabolomic, which means we are looking at the urine of these people and of the centenarians and offspring, because we collected many more that have been studied in this way, in order to identify metabolites which can suggest a fingerprinting, and in order to identify products of the gut microbiota that you can find, for example, in the urine with NMR. These are some of the data which just came out by our collaboration with Nestlé in Lausanne, and here you can see there are the bacteria, and here are the metabolites, and of course this analysis at the beginning is quite complex. These are, and you can have about 20,000 peaks for each NMR spectrum, and this is another, just to give you an idea of the complexity of the data, but believe me that some of the, there is a fingerprinting, and we can identify in the urine of the centenarians and the other people specific metabolites related to their gut microbiota composition. I think that this is a very promising way to look at the inflammation and inflammation markers. And of course, we are looking for this type of correlation between gut microbiota and metabolites in the urine. But of course, you can check in the urine a lot of other metabolites related to arachidonic acid cascade, carnitines, and so on. Epigenetic. Is known that epigenetic changes occur with aging. The literature is not so much. And again, we have studied 25 centenarians, 25 offspring of centenarians, and in comparison with offspring of non-long-living parents. So the offspring of centenarians have one parent is centenarians, and the other is long-living, more than 10 years of life expectancy for the Italian demography. The offspring of non-long-lived parents, both parents lived not more than the expected life expectancy for Italy, so which is about 70 years at that time. So they were dead before that age. So we try to have two very different group of people, and then young subjects. The population studies belongs to a much bigger study in which we collected recently 156 centenarians, 267 offspring of centenarians, and only 100 offspring of non-long-lived parents, which are very difficult to find, the most difficult. These are some of the pro-inflammatory mediators between the offspring and the offspring of centenarians and offspring of non-long-lived parents. And I would like you to give a look, for example, to this, which is very important. The centenarians have a very low IGF-1. The centenarian offspring have higher, but much less than the offspring on non-long-lived parents, suggesting that this is an important marker against many others. I have no time to go into details, and fit very well with the previous speaker. And in particular, we are now submitting a paper in which we have studied not simply the IGF-1, but the biological active IGF-1, because you know that IGF-1 can bind to several proteins in the blood. In general, the centenarian offspring are in much better shape than the offspring of non-long-lived parents. and the weight is is lower, waist circumference is lower, BMI is lower, the number of drugs assumed is lower, the number of myocardial infarction is lower, the stroke, cancer, hypertension is lower, and so on. Hand grip strength is higher, and the capability to perform some tests like chair stand is much better. So despite having the same age, the age of the parents makes the difference. And we have studied the global DNA methylation of this group with an anti-5-methylcytosine antibodies, and you can see that there is a decrease between young controls and centenarians, but there is also a big difference between the centenarian offspring and the offspring of non-long-lived parents. So there is a significant decrease in global DNA methylation levels with age, and the age-related loss of DNA methylation is less pronounced in centenarian offspring than in their controls. Then we looked at the ALU methylation, the non-coding part of the genome, and again you see the same and the big difference between with age and the big difference between the two groups of offspring. So a significant decrease in ALU methylation was observed in centenarians and offspring on non-long-lived parents. No significant change was detected in centenarian offspring, and so ALU methylation was statistically higher in centenarian offspring compared to offspring on non-long-lived parents. Then we performed a whole genome epigenome study. So we studied using this Illumina platform with 27,000 CPG, 20,000 encoding region, encoding CPG, and more than 7,000 repetitive DNA. Using this Illumina 27K platform, we studied the subject that I showed you. And in this case, here is reported the methylation of ALU-enriched sequences, and there is a decrease with age and a difference between the two groups of offspring. The mean methylation level of CPG repeats highly enriched in ALU sequences decrease with age, and there was a better preservation of DNA methylation status in centenarian offspring. This is the methylation value of the CPG islands, and again there is a difference. There is an increase with age, and a difference between great difference between young and centenarians, and the difference between the two groups of offspring. So the CPG sites within CPG islands were largely unmethylated, and the methylation level of CPG island increased with age from young controls to centenarians. And again, the average methylation value resulted significantly higher in centenarian offspring than in offspring of non-long-lived parents. There is also the distribution. If you look at the distribution, chromosomal distribution of these changes, you can check for changes related to aging, so the group of 70 people, and to longevity, and you can see that these are the chromosomes. Each bar is the chromosome, and there are some chromosomes which show changes more important than others. So it's not evenly distributed in the entire chromosome. This is very important because, of course, we still do not understand the significance, the meaning of this finding, but we are working on it. Using a very stringent criteria to look at the difference between the two groups of offspring, we arrived to a short list of genes and locus and loci, which are different in the two people, and this is the genes and these are the loci, and I am particularly glad that at least something came out from chromosome 11p15.5, because in this region we identified in previous studies several candidate longevity genes. Regarding the transcriptomic, what we have done is this is the paper that we have published recently in human T cells, and within the framework of a previous European project TCI, coordinated by Graham Pavlich. We studied the blood sample of 25 healthy male donors and in the T cells, and the groups were divided in 5 groups: young, adult, middle-aged, old, and very old, A, B, C, D. And what appears is quite striking is that the big clustering suggests to you that there is more similarity between the old, E, the E group, and the young and adult, AB, than to the middle-aged and old. And of course, there are many other data, but what is, I think, particularly interesting, the message that I would like to give you is that there are group of genes which have different trajectories with age. This is a group of genes which decreases in middle age and then increase. This is a group of genes whose expression increases and then decreases, and there is another bunch of genes whose expression is stable until 60 and then decreases. So the changes in gene expression profiling of different genes is quite complex. There are not monotonic trajectory. All the genes are increasing or decreasing. It's extremely complex. Some are decreasing, some are increasing, and there are groups of genes which follow these main trajectories. We are doing transcriptomic in human liver, in skin fibroblasts, and monozygotic twins, but I have no time. Some words for the genetics that we have done. I was the coordinator of a previous big, large project, GEHA, Genetics of Healthy Aging, in which we collected 2,568 90-plus seed pairs, so brother and sisters, couple of brother and sisters, which means familial longevity, because if at least 2 brothers or sisters arrive to age 90, suggests that there is some longevity in the family, and the same number of controlled for a total of 8,000 people. And we extracted more than 11,000 DNA here in Paris. It was done here in Paris at by Hélène Blanchet. And the GEA design allows to do linkage analysis in which you compare the couples of SIBs by couple of SIBs. And since there are younger controls, you can make association studies on genome-wide, GWAS. What we have done in the genome-wide linkage analysis用 an Illumina Infinium platform in about 2,564 seed pairs for a total of more than 5,000 subjects. The number of SNP was about 6,000. And when you do this study, you look for the coinheritance of chromosomal regions with the trait in families. It has for alleles, frequencies, and proportion of alleles shared identical by descent. Test whether siblings, the linkage analysis tests whether siblings affected by longevity, which is a trait, share more alleles transmitted to them from the same parents than expected by chance. And is very powerful, it's more powerful than association analysis, the GWAS, for identifying rare, high risk, but strong, powerful risk alleles. We have identified 4 linkage regions, resulting in a log score above 3, and the fine mapping of this region is in progress because they are quite large, but we hope in a short time to identify these new longevity genes. We have also done a piece of GWAS. GWAS identify common variants with small effect. It's the contrary. Common variants with small effect. Linkage is rare variants with a strong effect. And GWAS has been performed in 1,090 plus pairs, and the remaining samples are in progress. What I would like to share with you are the last data on the mitochondrial DNA, which is an integral part of this GEA project. We have studied not only the nuclear DNA, but we put a lot of energy to study mitochondrial DNA variability. Mitochondrial DNA variability can be either hereditary, and you know that the mitochondrial DNA is inherited by the mother, or somatic, which can occur in some cells of your body and of course are not inherited. In the first case, the hereditary variability, we are talking of haplogroups and subhaplogroups. These are haplotypes, in other words. In previous work, we have, particularly with the University of Calabria, we show that the mitochondrial haplogroup J is related to longevity, and that in centenarians there are higher levels of the C150T mutation in the control region. And the third finding that we have published is that there is a counterintuitively, there is a higher level of heteroplasmy in control region. Heteroplasmy means that in the same cell, in the same individual, you have two types of mitochondrial DNA, one which is mutated and the other which is not. Now we have studied 7,000 mitochondrial DNA samples, and we have published this paper that somatic point mutation in the mitochondrial control region are influenced by the genetic background, so are related in families and are associated with healthy aging and longevity. And this is something that strengthens and extend a previous investigation in which we have studied centenarians and their offspring, in which we show that the mitochondrial DNA control region show genetically correlated level of heteroplasmy in the leukocytes of centenarians and their offspring. So these are two big studies suggesting the same message. And indeed, heteroplasmy level in the loops is significantly higher in centenarians and their offspring. This is the study of 3 years ago. And the second study suggests that there is high and high correlated in the two members of 195 90 plus seed pairs from northern, southern Italy and Finland, so different countries. Not different because South and North Italy is different, but because Italy and Finland is different. Correlation, this is the correlation in the heteroplasmy. This is SIB1 and this is the heteroplasmy of SIB2, and you see that there is a beautiful correlation suggesting that in the two members of a family the level of heteroplasmy in the mitochondrial DNA is highly correlated, and so is probably genetically controlled. Now we have we have also more than 1,200 GEA complete mitochondrial DNA sequences. I remind you that the sequence of the mitochondrial DNA is 16,609, and so is a small DNA, but in any case this is one of the largest collection of complete mitochondrial DNA sequences in humans, and particularly in old people. And this is one of the last results. These are what the mutations, the somatic mutations in the complete mitochondrial DNA sequence that we found in the 377 90 plus SIBs and the 394 young controls from Denmark. Each point is a mutation. You can see that here in the control region there are more mutations as expected, but there are many others all along the mitochondrial DNA. How to interpret this data? It's not easy, because if you look at a single association analysis in which is any single allele significantly more common among the very old than in the young controls, okay. But if you suggest, if you hypothesize that two allele association, two SNPs, two mutations can contribute to the phenotype, the number of combination of the two alleles becomes very high, 274 million combinations. And if you have the idea that three SNPs, three alleles can contribute to the phenotype, there are 75 billion combinations. So it's computationally demanding, and we are trying to solve this point. So maternally inherited mitochondrial DNA variants are involved, but there is another side that I would like to share with you, and is that there are a lot of data that I showed you suggesting that the mitochondrial DNA variants are involved in aging and longevity. But what about age-related diseases. We have studied two major age-related diseases, Alzheimer and type 2 diabetes. This is a paper that we have published recently in in 936 Alzheimer patients and 776 cognitively assessed normal controls. And what we have found is that there is an haplogroup H5 which is different, sub-haplogroup, which is different between Alzheimer and controls, and which has a high odd risk. Another study that we have done recently is that on diabetic people. We studied the 466 type 2 diabetes patients and 438 controls. And all these people, 900 samples, were classified into 57 different mitochondrial subhaplogroups, thus reaching an unprecedented level of resolution. There is no other studies of this type in the literature. The most important finding is the following. When we compared the patients with the control, we didn't find practically anything. So we were really disappointed because it's a lot of work. But when we looked at more carefully to the diabetic patients and subdividing them in those who have complications and those who don't, things are much better. And I remind you that in the type 2 diabetes, oxidative stress and the insulin pathway and inflammation are highly involved, and that oxidative stress and mitochondrial dysfunction can play a major role because insulin resistance and diabetes can follow. And this is the paper that we have recently published in a few days ago. And the title is quite interesting. Mitochondrial DNA background might because the reviewers suggested that it was too strong, modulate. We had to have might, otherwise the paper could not be published. But anyway, mitochondrial DNA background might modulate diabetes complication rather than type 2 diabetes as a whole. And this is important because we can have, for the first time, a genetic tool to disentangle diabetes with and without complications. And these are the numbers, and you can see that we found a little bit a difference between the patients and the control in this haplogroup H1, but was at this borderline, despite the big difference is borderline because the Bonferroni is always there, but there are many other haplogroups which show big difference between the cases and which are quite interesting, and particularly what emerged is that H3, H star, H3, U3 and V, and they are correlated, H3 with neuropathy, H with retinopathy, about 1/3 of the diabetic patients have the retinopathy, U3 with nephropathy, and haplogroup V with renal failure, which is the last one. What is interesting is that looking in this, from this perspective to diabetes, if you look at the telomere lengths in peripheral blood between diabetic and control, no difference. But again, if you subdivide the diabetic patients with 1, 2, 3 or more than 3 complications, there is a great correlation between the telomere lengths and so on. So tell me when I have to stop, because so the magnitude of the effect of the decrease of the telomere lengths on complication is described by an estimated odd ratio of 5.4, which is not bad. Now, if I have 5 minutes, I would like to tell you something related to a hot topic. In aging research, there is a urgent need of robust and reliable biomarkers capable of distinguishing between biological versus chronological age, physiological versus pathological aging. And of course everybody is looking for these biomarkers. And I have the privilege to be a part of the European consortium called MARCAGE, which is examining about 4,000 people, collecting and examining 4,000 people for hundreds of parameters, and this European project is coordinated by Alexander Berkeley in Constance, Germany. But what I will propose you is a marker that is included in MARCAGE, but that we discovered a little bit earlier, which is glycomic. Glycomic, you know that an N-glycan, an N-linked oligosaccharide, is a sugar chain covalently linked to an asparagine residue of a polypeptide chain. The linkage commonly involves this type of residue, and the consensus peptide sequence is this one. Glycans can mediate a wide variety of biological role by virtue of their mass, shape, charge, and other physical property. It is important to emphasize that unlike protein sequences, which are primarily gene products, glycan chain structure are not encoded directly in the genome but are secondary gene product because they are produced by specific enzymes. In fact, glycans are synthesized in many different combinations by a variety of competing and sequentially acting glycosidases and glycosyltransferases. And this has been done in collaboration with Chiti Chen in Ghent, and they set up an ultrasensitive technique profiling to sequence the N-linked glycans using this DSA phase, DNA sequencer adapter fluorophore-assisted carbohydrate electrophoresis, and they need only 2 microliter of serum to do the analysis. This is the first paper that we published in Rejuvenation Research. We were the first to show, to suggest that the N-glycomic changes in the serum proteins during human aging is a strong biomarker. And in particular, what they do is to cut the oligosaccharide moiety, to mark, to label the glycans, and to desalylate, and to profiling using a DNA sequencer. And these are the main peaks that they can observe: peak 1, peak 2, peak 3, peak 4, peak 6, peak 8 and 9. These are the 2 most important peak 1 and 6 for aging, and peak 1 increased gradually with age, and peak 6 decreased gradually with age. And its ratio is the glycoage test. And you can see that peak 1 increases with age, and peak 6 decreases with age. And there are these are the peak 6, and you can see that the difference is the lack of these sugar hairs. And there are the galactosyl transferases which do the job to add this part, and the galactosidase which cut this to have the peak 1. And this is a striking correlation with age. We have used several centenarians here from 20 to 100, and you can see that there is a beautiful correlation. Then we have published another paper in which we suggested that is not only a predictor of healthy human aging, but also can be used in the diagnosis of hepatocellular carcinoma. And another one just came out, serum glycan profile during human aging. And you can see that in this case we have done the study on some cocaine syndrome patients, which are progeroid syndrome, and this has been provided by Jan Oymaker. And you can see that these are people of about 7-12 years, and their level are very variable, but much higher than the young guys. And another study compares Italians and Belgian centenarians. They are not so in Italian were 84 centenarians, and these are centenarians in Belgium, and there is some difference. And I have one of my grandmother was Belgian, French Belgian, but I discover now that there is a little bit difference between Italians and Belgian people. What is different, what is extremely interesting is this one. You know that Down syndrome, or trisomy 21, has an accelerated aging of the immune system of the brain. After age 30, in most Down syndrome subjects, a neurodegeneration starts, which worsens their previous handicap, mental handicap, and so Down syndrome is considered a segmental progeroid syndrome. We examined a 52 Down syndrome subjects of different ages from I think 13 to 72, because nowadays Down syndrome subjects are treated like normal persons. They are not discriminated as in the past, so they live much longer. And this is a problem because in this case, for example, of people of 60 or 70, their parents are either extremely old or they died, so there is a lot of problems. But apart from this, from a biological point of view, we studied Down syndrome subject, and in order to have the most suitable controls, we used as controls their siblings, which are rather age-matched, and another control is their mother. Of course, the mothers are older than the Down subjects. And look what the glycoage test tell us. These are female and males, and suggest that the Down syndrome have the glycomic tell us that they are much more similar to their mother than to their siblings, suggesting that their biological age is similar to that of their mother. So they are older, about 20 to 40 years older than their chronological age. These are the data, and these are the mothers, these are the Down, and these are the siblings. So this test is capable of telling you the biological age. We did also study on Werner syndrome, on cocaine, on Down, and this suggested, and centenarians. So the oldest one and those who have accelerated aging. I have finished. And this is just to tell you that we also studied the diabetic patients and controls that I showed you previously, and the data suggests that again, the glycomic, this glycomic approach, is able to identify peaks, in this case peak 8, peak 2, peak 8 or peak 3, which are correlated with the, not with the disease, but with the complications. So again, 3 types of tests: the mitochondrial DNA genetics, the telomere length, and the glycomic, which suggest that what we call diabetic are an heterogeneous group of patients, those with and those without complications, and that the biology of the two groups is quite different. This is my group, and I thank you very much.
So I think we've heard some very exciting current studies and studies on the horizon, and I'm sure there are some comments and questions.
It's Giovanni Mann from London, Kings. I have a question that you didn't touch upon, but you showed it in your introduction. And you were looking at in utero developmental priming of healthy aging. And my question to you is, in your centenarians, where you showed that the offspring from centenarians and long-lived parents had beneficial readouts, endpoints, do you think some of that was set during gestation, i.e. pregnancy?
Well, we don't know, unfortunately. Unfortunately, we don't know, but what I can tell you is that we are trying to look at, for example, twins of different ages, including very old twins, and we are recruiting blood from newborns who have been born about 1 kilo less than the 3.2 kg usual. And this means that during the gestation they underwent a variety of adjustment of the metabolic set points, and also to those who are born prematurely and kept the machines until they reach a weight which is compatible with life, with external life. And the idea that we have is that epigenetics is extremely powerful. I have preliminary data on the new platform which has been recently released by Illumina, which you can measure 450,000 CpG cytosine. Okay, so you have about 500,000 of CpG you can measure in each genome, and we studied 16 families, one Down, one mother, and one SIB, with this new platform of about 500,000, and the difference is enormous. And we identified, despite the fact that we analyzed not so many people, now we will analyze all the other, but also with such a small number of families, the Down are extremely different from their siblings, suggesting that the epigenetic fingerprinting at high density is extremely powerful. There are about 6,000, 7,000 CpGs, which are very different, and of course they are in part of the genome which is coding, other are in repetitive sequences in different chromosomes, so the analysis is just I would recommend epigenetics as a robust approach to identify difference. So this is the message that comes out from our data.
If I remember correctly, chromosome 21,
yeah.
which is very heavily methylated,
yeah.
is the area where FOXO3A genes are located, which control the insulin signaling pathway.
Yes, yes.
Is that right?
Yes. What we are looking, of course, is gene ontology and everything in order to identify not only single genes but pathways like the one that you suggested. The other point that comes out from this type of studies is that it can be very useful to approach an old problem, which is the X chromosome inactivation with age. You know that one of the female have two X chromosomes. One and they are randomly inactivated, okay? So when they are young, 50% is randomly inactivated. There are a lot of data in the literature suggesting that with age, there is a preferential inactivation of one of this X chromosome. And this type of approach can allow us to identify if this is correct or not with a very powerful approach, and if methylation or epigenetic is involved in this study. We have preliminary data which suggests that the age-related X inactivation in the offspring of centenarians is delayed.
Other comments and questions? Yes, sir.
This is John McGuire. I have a question. In mitochondrial genome studies, you do expression studies, have you been able to correlate the expression of mitochondrial